actually slow. It's about 2 to 3 minutes slow, so it's 4:02 right now.
Yeah,
Just to give you
4:02.
an idea.
Okay.
All right. Well, thank you all for coming and sitting inside on this beautiful day. I'm Jennifer Fuentes, and I'm here to introduce our presenter for today, Professor Anna Cavinato. She received her PhD in Analytical Chemistry from the University of Bari in Italy. She's a professor of chemistry at Eastern Oregon University, where she's been here and joined us since 1992. Prior to EOU, she was a research professor at the Center for Process Analytical Chemistry at the University of Washington, and an assistant professor in the Department of Chemistry at University of Memphis. She also received her postdoctoral training at Oak Ridge National Laboratories and University of Tennessee Medical School in Memphis, working in the area of protein purification and electrochemistry. For many years, she has conducted research using infrared spectroscopy, yes, to develop non-destructive methods for analysis.
Oh, it's not on. Maybe it's not
There
on.
we go.
There you go.
Better? Okay. Most recently, she's been working in the artificial DNA and development of nanosensors for bacterial detection. She's also received funding from NSF for curricular initiatives, including incorporating service learning experiences and developing research supportive curricula in chemical sciences. She's an active member of Analytical Sciences Digital Library, working with educators around the nation to develop and disseminate inquiry-based case studies and active learning resources in support of science instruction. Most recently, she was awarded a STEM grant from NSF to provide scholarships to financially needy and academically talented students majoring in STEM fields. Just as a reminder, if you have questions, raise your hands afterwards, and I'll come around so our live stream folks can hear us. Unfortunately, they might not have been able to hear me initially, but I'll turn it over with you.
All right. So thank you again all for being here this beautiful afternoon. When I was asked to deliver this colloquium, I kind of thought about different topics and how technical I could be. So I actually chose to go more into my teaching approach, but it's all permeated by research. So you will see that really throughout all the different classes, courses, particular lab courses that I teach, the approach is really rooted in research projects. So to give you a little bit of a background, I am an analytical chemist. So analytical chemists are they do all kind of different things. They are more of a service. Some people say we are like the servants to the chemist because we operate wherever an analysis is needed in a larger context. My teaching assignment is teaching analytical chemistry, which is really introducing students to the analytical method, and I will explain what that is in the next slide. But also there are a lot of components of statistics, designing sampling plans that are statistically valid and things like that. And then in the winter, we move into instrumental analysis, where I actually teach students how to use the instrumentation to conduct the analysis. And then in the spring term, what I'm doing right now, I inherited the third term of general chemistry. So that's where I really introduce the students through the lab activity to this idea of conducting lab inquiry in the context of a larger question. And of course, I do also research with students at different levels. So that's like my research students that work with me on specific topics. And I will touch about that at the end of my talk. So when I inherited these courses, they were really more in design in a traditional way, where labs were mostly canned labs. So a canned lab is where you already kind of know what the answer is. And of course, there is merit to that, but really doesn't really portray what a scientist is supposed to be doing eventually once they enter the workforce. So since about 2005, I've really kind of changed it. I constantly change. Every year, more or less, I do something different to adjust and to improve what I do. But a lot of the things that I'm currently doing are really an outcome of my involvement with professional societies, particularly the American Chemical Society Committee on Professional Training is a very important source of inspiration, I would say, on what makes the key component of instructing or developing a chemist, a scientist. So if you see, there are some things that probably apply to many other disciplines. Problem-solving skills, very important. Being able to search in the chemical literature, find scholarly work, base research on scholarly work, learn information management skills, so being able to use databases, being able to manipulate the data in all sorts of stuff, being able to conduct experiments in a safe environment. Very important communication skills, both oral and written communication skills. Team skills, because when they will go out in the workforce nowadays, all science is highly integrated. So a chemist might have to work right next to an electrical engineer or a computer scientist. So the idea of being able to develop these team skills and deal with people of all different backgrounds, not just in terms of upbringing, but of disciplines. And also ethics, which is what kind of brings in the component of community involvement in my research, because specifically CPC recommends that the curriculum should expose students to the role of chemistry in contemporary societal and global issues. And so that's where the component of research that comes from local issues, the local environment takes place. Another organization that also plays a role, it has inspired me, I would say, because it's not that I have invented any of this stuff, it's just that my exposure with people around the country, is the idea of developing a research supportive curricula, and specifically design undergraduate curricula in which students begin the scholarly activity from day one. So they are really being raised, if you want to say, with this idea that they are researching, and that might be from the simple introductory courses all the way, oops, there you go. I lost it, but anyway, all the way from from the introductory courses to their capstone courses. And of course, there is also kind of a personal engagement, because I am an analytical chemist. I really believe in the analytical process when I teach. And so I ask myself, do I really, when I teach my classes, do I really encourage my students to do this? So like if they are given a certain question or problem, can they really identify what the problem is, and can they really then find the methods to solve this particular question? Can they conduct the experiments and then eventually analyze data? So how can I engage students that way, and how can I portray my discipline in a broader context? So that's what I do. And what I'm going to show you is how I basically do this throughout the different courses. By the way, one way for finding projects that are suitable for students and that really somehow link them to the local environment is the idea of connecting to some community partners. So that, and sometimes the community partners are truly looking for some of that service from us. Some other times it's just an opportunity to have a project. So it's a great opportunity to engage students in these collaborative efforts and connect them to real world setting. But also it's really interesting to observe the students, because if you tell them that somebody else really needs the data that they are generating, there is some ownership in what they do, and so they really want to do it, and they want to do it right, because they know that somebody else is then going to look over that data, and it's going to make some sense out of that data. So that's service learning has many different synonym, but that's community partnering and things like that. I may have skipped a slide. Yeah. So I have a plan in my mind when I do this. These are my learning objectives. So I want to make sure the students have a question that they can inquire about, first of all. Also, I want to make sure that they have the opportunity to use analytical skills and use instrumentation all the way from the freshman year. So I've completely changed the Chem 206 lab to where students actually go into the instrumental analysis lab and can take advantage of industrial grade instrumentation. Some of them are maybe biology major, exercise, science. They may never again see those kind of things, but at least they've seen it once, or they've maybe developed some appreciation. And also, I want the students to collect their samples, whether it takes to go to the brew pub, as I will show you later on, but I always want them to go and get the sample. That connection with the sample completely changes the outlook, because it's very different to get a powder or something from the stockroom that doesn't mean that much, as opposed, say, to go to Marsh and collect the water samples and then do the analysis on those water samples. Again, everything, that connection is in part important because it shows, the grand picture of things, and also because from a realistic point of view, when somebody goes into the workforce, it's not the sample. There will be something they will have to deal with, and they will have to think about, how am I going to collect a sample that is really representative of what I will have to analyze. But then there is more, there is, wow, this is very sensitive. I'll be very gentle. Maybe, I wonder if the door, because the door is it in there? I think so. Maybe I'll be over here. Okay. So I'll start by what I do with my general chemistry class, and these are water monitoring projects. So when I started this, probably about 10 years ago, we used to go to N Creek, which is off of Hunter Lane, I think. Yeah, it's Hunter Lane. So N Creek used to be irrigation, well, it was changed into an irrigation channel and was eventually restored by a number of partners, including the Grand Ronde Model Watershed, the tribes, the Oregon Department of Fish and Wildlife, and so on and so forth. More recently, we go to Marsh, because Marsh, I don't know if you know that, but it's where the water from our water treatment plant is discharged. So there is an interesting ramification about, what happens to that water, what kind of nutrients may be present in that water. And so we actually go there with the class, and the way that the Marshall is laid out, basically the water treatment plants are over here, and this is the road that goes to Union. So if you turn on Peach Road, there is a distribution pond where the water is then, after chlorination is collected, and then the water kind of percolates through the marsh for about half a mile. So we sample at the distribution pond, which is it's not raw water by all means, because the water has been in the ponds that you can see if you travel along Interstate 84. But anyway, it's certainly the most raw of those, of the water. And then we sample it about half mile north. And then we also sample it at the fishing pond, because the fishing pond is not connected with the wastewater treatment plant. So the question that is posed to the entire class is whether there are really any detectable differences in water quality when the water enters the marsh from the distribution pond and then moves on throughout the marsh. So we do a number of different activities there. So we do a field trip. So we take each lab section does a field trip to the marsh. And at the marsh, we collect a number of data in the field. Use fairly rudimentary test kit. And then we bring them back to the lab. So you can see on site, we measure things like a temperature, pH, dissolved oxygen. We use hack kits to measure some important nutrients like nitrates and phosphate. And then we bring lots of water back, and for the rest of the lab, we look at all these different, these same analytes, but we actually use industrial grade analyses. So they'll use like an ion selective electrode to do nitrates. They'll do phosphate by spectrophotometry. They'll do metals using atomic absorption, and so on and so forth. And so this is an example of the kind of data. The numbers are pretty small in there, and the idea is not for you to really look specifically at those numbers, but rather what I was saying before, the ability of the students to gather data, to put them in some meaningful way to look at them. So here are all our different analytes, and here are, actually this is data that we collected over 2015, 16, and 17 in the different locations. Of course, this is a snapshot, so it's not that we can draw any major conclusion from this data, but the whole idea is to involve the students into something that is meaningful, and that they can, track over time, and at the same time learning those lab skills that I was talking about. So this kind of approach is then implemented in upper division courses. So for example, in Chem 321, which is the analytical chemistry lab, I work in collaboration with the US Forest Service. And so we go to Meadow Creek, and I'll show you a map of that. But at this point, the students are more familiar with the techniques, but now they have to worry about how do you sample? If you have an environment, how do you decide where to collect the samples? So we have a partner, Dr. Mary Rowland, that she's come to my class different times, and she's actually discussed what this project of Meadow Creek, which is a star key, is all about. So the US Forest Service project is to look at stream remediation. So Meadow Creek is being remediated for increasing the population of salmon, but at the same time with different grazing approaches. So that's different because normally in a restoration type of environment, grazing is not considered, but in this case it is. So you can see the different pasture what they have done. So this is Meadow Creek, and these are different pastures. So there are 5 different pastures, and we have actually sampled at the boundaries between pasture 1, 2, and 3. So our sampling plan, of course, was checked by Mary, because it had to make sense with the project, what the type of data that they want to be looking at. So you cannot really see very well because of the lighting, but if you look at this little part over here highlighted in pink, that's a fence where actually deer can go by the creek. There are other areas where cow can go by the creek. So, and then there are others where there is no grazing at all. Nobody is doing this work. There is no water quality monitoring that happens, because the Forest Service or the Oregon Department of Fish and Wildlife, much like a Marsh, they don't have the resources to do this kind of work. And so we go there again, and we look at the sampled water at the different pastures. So this is now, we go there with a GPS, so that we know exactly the location where we sample the water. And then again, we conduct some some analysis on site, and then we develop a much more comprehensive set of data for those pastures. So we have only done that for now 2 years, just 2016 and then 2017. Again, you know, this is mostly baseline, because I think that the grazing only started like in the fall of 2017. So this is more baseline data. So it'll be interesting over the next few years to go back to those sites and see whether really we can see any differences in concentrations of nitrate. You can see that there are some differences across the pastures. So there are some areas where there is more nitrate, but the amounts are very small, because in reality you would have to have 10 parts per million to worry about it. So the quality of the water in reality is pretty good. Then this fall, for the first time in the same class, I introduced yet another experiment. So after we go and sample the water and we complete some analysis on that water, we switched gear, and we actually worked on a project with the ODF&W pathology lab to look at antibiotics in fish. So this is a new project and stemmed from the fact that fish that is raised in hatchery, because of the condition in which the fish is raised, require use of antibiotics. So the fish are routinely injected with different types of antibiotics of the macrolide class of antibiotics. And the reason why ODF&W is interested in this is because they typically return the carcasses of salmon into the streams for rebuilding again habitat for fish and adding nutrients to the stream. So the question is, if these fish, they are all pumped with antibiotics on a routine basis, and then they are put back in the stream, is there an issue of releasing antibiotics in the environment? So there are no studies of what is called the depletion studies of these antibiotics in fish tissue. So, of all people in the world, my students were tasked with learning about how do you go about finding antibiotic in fish. So this is a very complex project. This is not an easy project at all. And we started focusing on erythromycin, and actually now I have some research students that are looking into tulathromycin that is sold as Draxin. So this is where now they are really starting to become more independent. So if you see, I kind of guide them, they have the blanket in Chem 206 because I give them the experiments, the procedure of the experiments are given to them, they are known to them. They just needed to put this picture together. They needed to communicate to form this big picture. They have to do the analysis well. But now, the students in this class, they are actually tasked with researching the literature to find out what method to use, because I don't know what method to use. So they have to tell me, they have to research it. So that's where things get more complicated, because they have to research the literature, so they have to understand that they are dealing with this is the chemical structure of erythromycin, this is an example of a macrolide. So this is a big molecule. And so to deal with a big molecule like that, there aren't too many pieces of equipment that can be used. But so they have to keep that in mind, what they are dealing with, the chemistry of what they're dealing with. They have to keep in mind what we have in our lab, because unless President Insko buys us more instruments, we only can do so many, can only do so many analyses. So they have to choose the actual method of analysis and the equipment. Then they have to figure out all the chemicals that go into doing the analysis. They have to think about how are they going to prep the samples for analysis. This is a major issue if they were in any other industry, like whether it was an environmental lab, whether it was a pharmaceutical lab, a food lab. You can just take a sample and put it into the instrument. There is a lot that goes into the preparation of that sample. And of course, then all the other aspects. Sometimes we don't get to interpret the data because we don't have enough time to really generate the data that can be interpreted. But nonetheless, the whole exercise is just incredibly valuable. So, for example, for this one, we can't really see the chromatograms at all. I think the light is so, maybe can you turn the lights off for just a second? Yeah, so for some reason they don't show up in this PowerPoint. You have to believe in me. Okay, that's all right. But anyway, I'm sorry, because I had some really nice graphics, but for some reason they're not showing up here. But anyway, so the students came to the conclusion that they could use liquid chromatography with UV detection. And by the way, if you don't know what chromatography is, you may have done paper chromatography, like if you take a marker and you put it on a coffee filter, and then you put the coffee filter in water, and all the bands of different color, the pigments separate. Well, that's a very cheap way of doing chromatography. Interesting. Anyway, the slide is not there. But anyway, we use a much more involved, of course, a type of much bigger instrument that is really a very fundamental piece of equipment that anybody that goes into either environmental, forensic, or pharmaceutical will be working with. So the ability to operate an instrument like that is really important. So they had to learn about sample preparation, how to extract the antibiotic from the tissue. So fish tissue is really nasty to work with, so they had to cut all this fish yuck stuff and put it in put it in test tubes and use different solvents and so on and so forth. One very important concept in any type of chemical analysis is the concept of calibration. So you cannot use an instrument without calibrating that instrument with set standards. So these are solutions of known concentration. So we were able to calibrate very beautifully our instrument for erythromycin all the way to about 0.25 micrograms per milliliter antibiotic. So this is an ongoing project now that we're trying to tackle with a smaller number of research students. But you can see these were actually students that worked with me last summer. They started this project, and then that's Tony Capps and Sally Gee from the fish pathology lab. They are very gracious because I go and pester them all the time. And then Gabby Rodriguez, she was a Project Seed student, so she was actually from high school, and she was doing liquid chromatography in high school. That's really awesome. And then you see Cassidy here at the ACS meeting in New Orleans. She presented some of the preliminary results that we got on the fish, on this fish project, and there is somebody interested in the poster, I guess. And then when we get to the winter time, it's time to go down to the brew pub. And so this year, for the first time, I decided to conduct a field trip to Side Brewery, because we used to work with, so one of the experiments that I do in instrumental analysis is to measure polyphenols in beer. So polyphenols, it's a class of compounds that are characterized by these structures, and much more complex structure. You see some of them here, the three polyphenols that we analyze, but there are many, many others in edible plants, and they come in the beer. They mostly come from hops, and the husk of barley. But this is interesting because, again, it connects to a local establishment. It's something that they connect to. Every little body can connect to beer. We don't drink it, because rule number 1 in a chemistry lab, you never eat or drink your experiment. So that is always said from day one. But anyway, polyphenols are actually really interesting in beer because for one, they affect the flavor. So they come from hops, so they affect the bitterness of the beer, and overall the bouquet, the flavor, what you can smell from the beer, well, smell and taste in beer. To a certain extent, they can stabilize the beer over time, but if there are too many polyphenols, they will actually interact with the proteins in beer, and then they will create haze. So if you see a hazy beer, that might be due to that particular issue. So there are many, many polyphenols that one can analyze, but we chose to analyze these 3. And so we use, so this is our liquid chromatograph, the same that is used for the fish analysis. But in this case, we actually used the Space Scottish Ale. And what was really different in this experiment was that we actually looked at the polyphenols through the process of making beer. And I have yet to find a paper where this is described. So people describe the polyphenols in beer, but they don't necessarily see how they change to the process. And I'm not really sure what the significance of that would be, because I'm not a beer expert. But I have talked to Nicholas, who is the brewmaster, and he's very interested in this. So we went there, and we actually got beer sample, very small, like 25 milliliters. So nothing that you can worry about. And so we got that beer from the wort, which is where the initial cooking of the barley happens, and they start adding the hops. And then we also got beer from the fermenting tanks, and then the finished product. So we basically followed the composition of those 3 polyphenols, as well as a total polyphenol analysis done by UV visible over these 3. And so again, you can see how this is out of my students' papers. So they calibrated for caffeic, peracumaric, and ferulic acid. So they need to know where those are. We do need a mass spectrometer desperately for that instrument. So if anybody wants to buy me a mass spectrometer to hook up to the HPLC, I will be very delighted because it would make these things even more so interesting and easy to determine. But anyway, you can see these are like the standard that they inject, and then if this is like the time that they elute, there is a column inside the HPLC that separates the different compounds, so they come out at different times, much like I showed you the little picture of the bands in the paper. It's the same idea. So we inject a sample, and then this sample travels through this column pushed by a solvent. And so every time a peak comes out, like this one, there is that is representative of a compound. And you can even see, like we injected the smaller amounts and bigger amounts. And then this is actually a chromatogram collected on a sample that came from the water. So you can, if you look at the the number are very small there, but if you look at what is called the retention time of this peak, and then you go look at the retention time, you will find those peaks in that chromatogram. And so these are some really kind of interesting results because what we see is that the polyphenols actually increase as the process of the beer goes on. So this is the wort, the fermenting, the finished beer. This is a total polyphenol analysis done by UV visible spectrophotometry. But then when we look at the polyphenols individually, the caffeic acid pretty much stays the same. And then the paracoumeric, which is this lighter blue, actually tends to decrease, and then the ferulic increases. So I don't really know yet what that means, but these are all data that the students collected. These are averages of all the analysis that the different groups actually did. So I also want to say that with all these activities that we do, there is always, different level of assessments. So very important is their lab notebooks. So their lab notebooks are becoming increasingly structured like a actual technical report, like a paper. So by the time they are in instrumental analysis, they are literally writing a lab report that looks like a journal paper with all the different components of a paper. Another thing that I have them do in upper division classes is that, like for the fish, they literally had to write a proposal. So they had to write a proposal of how they were going to go about the development of the method. So they had to research the literature, and then they had to write down what the analysis was supposed to be, what kind of materials they needed to have. That's a very difficult task for them to do. It's really pushing the envelope, often frustrating the students because it's like, we have no idea. But it's good for them to be looking at those things. Sometimes we have a presentation to community partners, so we have done that with the Grand Model Watershed. Conferences, presentations, including national meetings. I always like, especially at the lower division, students write a reflection paper of what it meant for them to do the lab that way and to actually be involved in a research project. So they reflect on their own learning and how was that different from other experiences, what worked and what did not work. And then, of course, we have all the way, to articles that are published in the Eastern Oregon Science Journal or peer-reviewed journals. So I don't have very much time. I just wanted to spend the last 5 minutes of this just by talking about the research that I actually do now with the students that are doing the capstone. That's Chem 401 research. It doesn't mean that they're necessarily upper division students. Some start when they are freshmen. But we typically operate in chemistry. We have teams of students that work around the project. So there is always like a senior member that kind of carries on, and then other students that learn from other students under our guidance. It's just not Colin, Ron, Colby. We all use the same approach. So this is a project that I've actually been working for quite a few years, and you might have heard me talk about this before. A lot of the research that I've done over the years is around fish, because I'm fascinated with diagnostics. And I've never been close to a hospital where I could do diagnostics on human beings, so I guess fish is the next in line. And it's great because we have hatcheries around here and lots of health problems. So one of them is bacterial kidney disease that can be quite pervasive and can actually kill like thousands of fish if happens in a hatchery setting. And so to this day, the test, so the actual disease is caused by a bacterium that is called the Renibacterium salmoninarum. And this bacterium produces, the reason why I bring this up, it produces a protein, an extracellular protein, that it's called the P57 or major soluble antigen, that is suspected in the virulence of the bacterium, and it's shed in water. So what I've been working over the years is to try and develop a bacterial sensor that replaces these other methods of detecting the presence of the bacterium that are much more lengthy in time. So it's not something that you can do right away. So if you have to culture cells or do other tests, it's a lot. It takes maybe days before you can ultimately have an idea whether the infection. By then, the fish is basically dead. So I've seen this at. There is a very large amount of research that is done on artificial DNA. And so the idea in this sensor development is to use aptamers. Aptamers are short sequences of artificial DNA that can actually operate as the antenna for a sensor. And I'll tell you a little bit about the sensor later on, although I mentioned here potential application would be to use nanoparticles, so to create nanosensors. So the idea is to, as I said, use this artificial DNA, and we actually now work on a template that only has 4D base pairs, while these other parts over here, these are related to the amplification of the DNA. So they are actually what are called the primer zones. But without getting too technical, the idea of isolating, the whole idea is to start from a very large pool of DNA, artificial DNA molecules, and react them with the protein, the molecule, the MSA, major soluble antigen that is spread from the bacterium, and isolate like a needle, a true needle in a huge haystack, those strands of DNA that bind to the protein. So we have worked on this now for quite a few years because it's a very, very challenging project. We started from this large pool of DNA. We reacted with the protein for a while. Then we actually, more recently, use graphene oxide as a way to remove DNA that is not bound to the protein. And then we release that DNA. We use polymerase chain reaction to make many, many copies of the DNA, much like they would do it in a forensic lab to find, somebody that is some DNA found at a crime scene. But ours is artificial. And so then we have to regenerate a single-stranded DNA by another procedure where we use some streptavidin, and we actually label the DNA with biotin and use the streptavidin biotin bond to link them on some beads and then elute them and recover. Basically, we are recovering the single-stranded DNA, and then we put back into this cycle to enrich the pool with the aptamer that binds to the protein. So finally, last spring, after about 3 years of doing all this, we were able for the first time to sequence a pool of DNA. So John Reinhardt was instrumental in doing that because I really don't know how to sequence DNA. So he helped my students to do that. And so we were able to, and it's very unfortunate that the graphics are not coming out as vivid in the slide. But in any case, we were able to determine the sequence, and then we put the sequence into a program that determined the structure. So that's what those aptamers look like potentially, and so then they bind into a special site of the protein. And so what we are doing right now is actually determine how tight that aptamer is binding to the protein with some binding assays. These binding assays have proven to be very challenging because normally they are done using radioactive compounds, and we cannot use radioactivity here. So we had to reinvent the whole thing. And so we were able to develop a strategy. I was very lucky to run across a graduate students from Taiwan in Washington, D.C. last summer. And I saw that he was doing this in a different application. And so I was able to, actually, we were able to implement, and we are currently working on finding the constant of equilibrium or the affinity constant of these aptamers with the protein. So if the affinity constant is good, that means that these aptamers will be able to find the protein potentially in the water. So then we have to find a scheme to develop the sensor. And one potential scheme is to anchor that DNA to golden nanoparticles, because the nanoparticles change color, whether they are dispersed or aggregate. And so if they find the protein and they become aggregate, then they change color. They change from a bright red to kind of a purple blue. So that's the next step of this research, will be to actually move into the sensor development if we ever find an aptamer that binds tight enough to do that. So very challenging. So I'm come to my conclusions. I hope that I have kind of told you the journey that we go through, basically by using laboratory activities where students really experience a research-like environment from their freshman year. Projects are chosen to illustrate topics that are relevant to this community, so it's relatively easy to interact with those that treasure the research that we are doing. Students are increasingly asked to research and implement a lab procedure and work independently. That's my goal, to kind of move them more and more towards independence and use lots of written and oral communication assignment things, because that is the key to be a successful scientist. You could be Nobel Prize, but if you don't communicate, if you don't let the world know the work that you're doing, then it's not going anywhere. So that's a very important skill to gain. And with that, I have lots of people to thank. My students, for sure, because they go through me. This is Haley Breen. She's one of the osteo scholars, and Jessica Carter in New Orleans presenting the work on the aptamers. So anyway, I will turn it to questions if there are any. And thank you again from listening to me for 50 minutes. Thank you.
So feel free if you have questions. I'll bring the mic right over.
No question. I scared off.
So.
Question.
Sorry, Nancy, get you first.
Could you talk a little more about how you think about that Research 401 class, especially since you have multiple levels of students in there? How do students migrate in? Are they able to take it repeatedly?
Yes, so absolutely. So the capstone at the 401, for one, research is a requirement for the chemistry, biochemistry degree. So that's built in our curriculum. But we really have a very research supportive environment. It's absolutely all of us. It's not just me, it's all of us that kind of nurture that environment. So in any case, whether students are doing research with me or Ron or Colin, some of them may come in as freshmen. And so because we have teams, usually there may be 3 students in a team, sometimes 4 students in a team. So the ones that come in are learning from the other students. Of course, we are always there to monitor what they're doing and provide guidance. But there is that peer interaction that is really important. And so then as students graduate, the ones that are coming through the ranks, they move on into more of a leadership position. So yeah, so it's kind of a revolving team, I would say.
I have a couple questions. How much is a mass spectrometer?
I knew it! The mass spectrometer is probably $70,000.
What do you think, Sean? You want to give up your microscope?
Okay. Let's arm wrestle.
No, more seriously though. One,
that's serious.
Yeah. Thanks for what you do. This is fantastic. I'm curious. I wasn't here for the very start, but when I read your summary, you said you've shifted this direction to do this more.
Right.
What I'm actually curious about though is we hear a lot about millennials and millennials needing to be handheld through a lot of life anymore, it seems like, or things being given to them. And you're really integrating an inner and independence into your teaching process. Has that been more of a struggle as the millennials have entered into college, or have you been able to maintain that?
I cannot say that. I don't think so. I would say that the struggles that they go through are pretty much the same that I have encountered over the years. It's a very different, it's a big shift in paradigm. And so one of the things that I think it's really important is for the teacher to explain the rationale. Because many of the things that I do could be interpreted like what she doesn't want to teach. She doesn't want to. But it's so important, it's the active learning type of approach. So I always explain to the students, you're doing this because my goal is to bring you over there. But I don't know, I would be interested to hear Ron and Colin on that. Have you seen differences in the ability of students to do independent research over the years?
Sure. I don't think that the students that self-select to do research have changed.
Yeah.
I think
That's a good point.
that there is a bulk shift. If you want to label them as millennials and whatever that means, great, you're baby boomers and whatever that means. But the people that want to do research, they're driven by the same things. It hasn't changed in the decades that I've done this process. Colin thinks as far as.
I probably the same thing.
It's independent. I think it's independent of the time. And I think those students also overcome whatever K through 12 nonsense is going on at the time. And in spite of whatever's happening there, they still manage to sort through the process and excel. Would there be a different number of people doing that if things were different? It's hard to say.
And of course, there are, all kind of a gradient of skills and abilities for them to operate. I mean, like the student that did the DNA synthesis, the DNA sequencing, Brian Mandela, he was, for all purposes, he was like a graduate student in my lab. Absolutely independent. I would go to him and say, okay, Brian, what are you doing today? And he would tell me. And I'm not kidding you. And that was phenomenal. Not everybody's like that. Not everybody. There are different skills. So. Oh, yes, Kathy.
Have you been doing this long enough to get feedback from your students, both
yeah.
at the end of the semester and then later on when they go off in life to come back? Does it help them get jobs? Or
Yes,
what do you
yeah,
hear?
absolutely. So the skills that they get by doing something like that, they are really, I mean, it depends, which direction they go in their career. Those that have gone on to work in labs, absolutely. They send me messages. Some ask me questions through Facebook. Thinks they're still in communication with me. So yeah, definitely. And then the reflection paper that they do in Chem 206, that's also another vehicle of determining whether that works for the students. And that's always in general, the feedback is very positive. And again, because they get to go and do a field trip, they are outdoor, they connect to the environment. They're not just, the key is, they're not just doing an analysis that is isolated and is just repeating something. They're doing this analysis in the context of a large question. Each of us would be, I would imagine everybody would be more engaged because you're looking at this larger picture. But the skills, the lab skills are really fundamental. 1 minute.
So as you've integrated more active learning into your classroom and such, from an instructional standpoint, what's been the most challenging in making that transition?
Part of the challenge is that you have to let it go. So if you have this kind of approach, you cannot cover as much material as you would do if you were just spewing out information to the students. So it's a question of quality versus quantity. That's kind of hard when you're trained to say, oh my gosh, I have to cover all these XYZ things. In reality, what they're doing though, and these are conversations that I've had at national level with colleagues around the country, they say, remember, it's much better for them to go and look at this entire approach with one thing, because whatever they learn there, they can transfer to something else rather than do like a survey. But that's hard, and it's also hard in the actual classroom setting, because I do a lot of class activities, but the class activities take a lot of time. So there is content that is sacrificed, but in the grand scheme of things, I don't think that that's a loss. But for me personally, that was a challenge. Then there is also the challenge from the student standpoint, because many of them, like in 206, so this is in general chemistry, they come from an environment where they are just taught, they just sit there and say, well, do we have to do this? Yes, you have to do this, because I always say this is like learning to play an instrument. You can listen to somebody playing the instrument forever and you never learn to play the instrument, so you have to actually do it. But yeah, there are challenges. And one key is to always explain the rationale behind. So if you tell the students, I'm doing this because I want you to get there, that mitigates some of the frustration at times. So anyway, I think we are out of time. So thank you. Thank you very much.